And thank you for the invitation to come out and tell you a little bit about what I study at the University of Hawaii. I've only been here for a few years, but it's a fantastic place if you're interested in understanding the evolution of visual systems. And particularly for me, I study the evolution of the visual system in mantis shrimp. And there are lots of those around to play with here. So it's been fun for me. And what I want to tell you about today is first just introduce some of why the ocean in particular is a great habitat and all the different sorts of visual systems you can find there, and then tell you a little bit about the sort of work that I'm doing with mantis shrimp. So I wanted to start off with vision in the ocean, and just people who live here are very familiar with how colorful and vibrant shallow water habitats and coral reefs can be. They're really colorful places, there are colorful animals, and so that suggests something about the visual systems that animals might have in those environments. So if we think about light in the ocean, we think about light as a scientist, I think about it in terms of wavelengths. So how long are the wavelengths of light? And that then translates into color. So here is a visible spectrum that we get from the sun. So at the surface of the ocean, we have the full spectrum, all the way from ultraviolet, the shortest wavelengths, down 300 nanometers, up to red, to 700 nanometers and beyond. So at the surface, and these shallow coral water habitats have this full spectrum of light, and they're very colorful places. However, the ocean, as you go deeper in the ocean, it attenuates light due to scattering and absorption, and very quickly, within the first few meters, as you go down in the ocean, you lose both ends of the spectrum. So you begin to lose the ultraviolet light and you lose the red light, and the deeper you go, the narrower, the less wavelengths of light there are. So you're left with, by 200 meters, really only blue light. Which is one of the reasons that it's such a blue place, the open ocean. Another feature of the ocean and the open ocean is as you increase your depth, you also lose intensity. So not only are you losing parts of the spectrum, you also lose how bright it is. So you have less light to work with. So it's across this really dramatic change in light environments, and animals live all across this, from the surface all the way down to past depths past where light penetrates. And this variation in light environment has led to the evolution of a wonderfully diverse set of visual systems that are adapted to the specific needs of each animal. And so just to introduce you to some of the really weird and wonderful visual systems that you can find in the ocean, I want to introduce you to some of the animals that perhaps you haven't seen before. One feature of this attenuation of light to only blue wavelengths, and also decreasing the illumination with depth, is that animals can take advantage of this. So a researcher at Duke named Sanka Johnson did a study looking at the coloration of animals with increasing depth in the ocean. And he found that in surface systems, up here where you see a lot of silvery fish, as you get a little bit deeper, you see a lot of things living in the open ocean that are transparent as a means of camouflage. But the deeper you go, the more you start to see animals that are red. And this again takes advantage of that very predictable way that light changes with depth. So if you remove the red light from the environment, then those things that are red become camouflaged. There's no red light, so being red is as good as being black when you get deep enough in the ocean. Particularly where light begins to become very dim, you get really unusual adaptations in the visual system. Animals down there where there's very little light left try to maximize what light is available. And so you get these really enlarged eyes, or really bizarre eyes. This is called a fish called a barrel eye. One of the weirdest sets of eyes I think I've seen in a fish before. The entire top of the head is transparent, like a window, and its eyes, these are its olfactory system, its nose. The eyes are these green balls here are the lenses. So it's called a barrel eye because there's a barrel with a lens on top that sticks straight up and looks through the transparent top of the head. So that's a pretty bizarre and fabulous visual system. Moving more to things that I tend to study, the invertebrates. This is an ostracod. It's a type of crustacean. It's called a Gigantocyprus, and they can get as big as a golf ball, ping pong ball size. And they have, again, to try to maximize what little light is left in the ocean, they have these giant reflectors. So think about a headlight. So if you shine a light at it, you get that light reflected back. And so to try to collect as much light as possible for their visual system, they've built a set of reflectors behind their eyes. Another really interesting group that has lots of weird visual systems is a family of amphipods called the Hyperiidae. This is one example. I like it because people claim that it was the inspiration for the Alien in the Alien movies. I don't know if you can see that resemblance. But if you look here, there's these two black spots on each side of its head, and so it actually has 4 eyes, 2 that point up and 2 that point forward. So you see this whole vast array of adaptations in the ocean as animals evolve to whatever particular tasks that they need to survive, to find prey, to find mates, to avoid predators. And in the open ocean, that can be a difficult task, particularly when there's very little light left. These 3 animals are just 3 examples, some of the really weird visual systems you find in the ocean. Some others that I wanted to point out, we tend to think of visual systems as being associated with the head. So because we have a big head with 2 eyes up front that we see the world with, we tend to identify eyes as things that in animals that are similar in the head to a pair of eyes for animals to use for vision. But there are lots of animals that have eyes that are not associated with a head or a brain. And so I just wanted to show you a few examples of those as well. And these are things that you may see if you spend a lot of time in the water in Hawaii. So one is the scallop. If you look at the scallop along the edges of the mantle here along the shell, each one of those blue dots is an eye. And so this is an animal without a head, but still has many, many eyes, which it uses to perceive and respond to the world around it. Another example that I quite like, and in collaboration with my friend Mike Bach at the University of Sweden, have started studying some of the eye evolution in groups of worms. So perhaps you've been snorkeling and seen Christmas tree worms out in the coral. And then if you have, you know that they're very good. As soon as you get close to them, they pull into their tube and disappear. One of the ways that they're able to perceive you, if we zoom in on these 2 in the corner, is that they actually have eyes at the base of those spiral branchiae. So if you look here, they have 2 banana-shaped orange eyes that are hiding under that Christmas tree structure. And worms in general are a really interesting group to study eye evolution because it seems like every group of worms has independently evolved a solution to needing an eye to avoid predators. So here's 2 more examples in fan worms, a different family of worms. So these are the Sabellidae. And on the left, you see Bispira, and each one of those black dots on these feathers is a pair of simple eyes. And then on the right, this is a Megaloma interrupta, and it has evolved a set of periscopes, essentially, 2 eyes up on stalks above the fan to detect predators with. And in this entire group, the eyes are distributed across their feathers, which both serve for feeding and for breathing. So again, another animal that has eyes distributed across unusual parts of the body. And that's really advantageous for this group because if you're sticking your gills and your feeding tentacles up into the water, that's the most vulnerable part to predators. And so you want to have a really good response system to pull back into your tube, which is your only mode of escape. I think the final sort of animal I want to introduce you to in the tour of weird eyes of the ocean are the box jellies. And these, again, you have a question.
Yeah, those eyes, they just detect light and dark, or they detect motion?
The eyes in the fan worms?
The previous in the worms.
In the worms, they are very good as shadow detectors. So it's a combination of contrast and motion. So the box jellies, which you may hopefully haven't run into here, but they are around. People get stung on a fairly regular basis, particularly around Waikiki. The box jellies, even though they don't really have a head or even a brain, they have a nerve net. They have 4 structures, one on each corner of the box. That's called a rhopalium. And each rhopalium has a set of 6 eyes in that structure. So in total, box jellies have 24 eyes. That's a lot for a jellyfish. And people have done studies on some of the box jellies that live around mangroves, and they think it's to help navigate and keep them within the protection of the mangrove forests. So they're not really good at resolution, but they're good at, again, contrast. And you can also see that the eyes are all pointed in different directions and are of different sizes. So they have eyes for different visual tasks, most likely. So with that as a brief introduction to the ocean and the light environment that marine animals are evolving in, and all of the wonderful visual systems that you can find there, I want to now move into talking about stomatopods, or mantis shrimp. And these shrimp are the species that I mainly focus on. They have a really wonderful and complex visual system, which I'll explain to you in just a couple of minutes. This slide shows you what a colorful and diverse group it is. It is an order of exclusively marine shrimp. There are right around 500 species that have been described so far, and they vary in terms of size and color, as you can see from the examples here. Just to give you a close-up of some of the species you might find here in Hawaii. There are about 20 species described from the Hawaiian Islands. Here's just 4 of them that I've run into anyway. Pseudosquilla ciliata, and Gonadactylaceus falcatus. Those are probably 2 of the most common. They're shallow water. If you see something out running around the reef, it's probably one of those. Odonodactylus brevirostris, that lives at about 50 or 60 feet. So to see those, you would need to be diving. Poiosquilla, these are tiny, tiny, and they lived in sand, so they're just a couple millimeters long. But we think we have a new species here, so we've upped the number of stomatopods in Hawaii by at least 1 so far. You may have seen this one. This is the largest species of stomatopod, and we also find it here in Hawaii. Its scientific name is Lysiosquilina maculata, and these get quite large, so 1.5 foot, lobster size.
Could you just briefly give the characteristics of a stomatopod?
The characteristics of a stomatopod, what makes it a stomatopod? So the most characteristic feature of the stomatopod is the group is, unlike other crustaceans, they are obligate predators. And so they're called mantis shrimp because, similar to a praying mantis, the insect you may be familiar with, they have these front raptorial appendages that they catch prey with. And in a slide or two, I'll show you how those work. But they're known for being very aggressive. And other names, people call them thumb splitters, because if you stick a finger in a hole that they live in, they are more than happy to punch it and split your thumb open.
Are they edibles?
These big ones, yes. People around the world do eat them. So this one does live here in Hawaii, and I'm always looking for it. It actually is very hard to find because it lives in sand burrows, so you almost never see it. If you do, it'll probably look like this. Here's one sticking its eyes out of the top of its burrow. But they never leave their burrow. They mate for life, and they only come out to catch fish. So stomatopods, to give you a little more information about the group, they are a culturally important group, particularly in the Pacific, not so much in mainland US. People aren't as familiar with them. But there are many places in Asia and the Pacific where they're eaten, where you can find stomatopods sold in fish markets. You can get stomatopod sushi if you're in the right place. I've been told that stomatopods were the original shrimp scampi. So if you go to Italy at the right time of year, you might have a stomatopod scampi. Also, so their raptorial appendages have been used for jewelry. Particular parts of the Pacific that they've been used to make weapons with. And then also they've been recognized as culturally important just in...
Where is that? Republic Summer, whatever that is.
I don't actually know. This was given to me by a friend. So if someone knows, I would love to know. Someplace that values stomatopods, so it's a good place as far as I'm concerned. So there are multiple places that have recognized stomatopods as something that is important to the local community and the local culture. Mantis shrimp are perhaps most well known. A few years ago, a well-known online cartoonist called The Oatmeal did a cartoon about the mantis shrimp that really made the general public aware of this group. And he wrote a comic called Why Mantis Shrimp Are My New Favorite Animals. And it's all about this perception of how mantis shrimp are these really violent creatures. And it is true, they tend to hit first and ask questions later. He described them as creatively violent and the harbinger of blood-soaked rainbows. So because they are so colorful and so violent, they tend to capture the imagination of the public. So as I was telling you a little bit about their, the easiest way to distinguish a stomatopod, if you happen to see one, are their raptorial appendages. So these are the appendages they use to catch their prey. Species come in two different types of predators. They can either be smashers. So if you see this arm here, here's just that arm. They're specialized for hitting things very hard. So they tend to eat hard-bodied shells, snails, things that they have to crack open. That Lysiosqualina, that really big shrimp that I showed you, catches soft-bodied prey. So these are spearers. So they have raptorial appendages with spines on them. And so they will come bursting out of that burrow to catch things as they swim overhead.
Again, what did you say that top is?
So this is the raptorial appendage. So this is just that hitting arm. So that's this part here. Sorry?
It looks like two people kissing.
No, that's just the raptorial appendage. This is the part that they hit with. So you can see it's enlarged and armored to protect themselves from cracking their own arm when they hit something really hard. To give you a look at how this works, I have some videos. The first one I want to show you is from the smasher type. And this is recorded at 5,000 frames a second, so it's going to be slowed down tremendously. And it's hitting a snail shell. So that flash of light you saw is cavitation. So they're moving so fast, they are creating vacuums in the water. And so they not only have one of the fastest recorded animal speeds in this strike, they move at the speeds on par with a .22 caliber bullet, so very, very fast. They've been measured as having 250 pounds of force when they hit, so they also can hit very hard. And then they also are moving so fast through the water, they create this cavitation, which is also a secondary, very damaging process. So they're extremely powerful predators, and you don't want to be hit by one. It hurts. I can verify that. To give you a look at how the spearers attack their prey, there it was. Everyone see that? See that again. But you can get a better look at that. Here again is a very slowed down video. So you can see it coming up out of its burrow to get that piece of fish, and it will give you an idea of the reach they have with their raptorial appendages. So, like I said, you probably will never see one of these because they tend to hide, but when they do want to catch something, they're very effective at it. So that is a little tour to the introduction of mantis shrimp. and some of the diversity that you find in the group. I want to move now to telling you a bit about some of my work and work from many of my colleagues who have been studying visual systems in mantis shrimp for over 20 years, and why they're a really unique animal, what's unique about their visual system, and what we know and what we still are trying to figure out. So before I tell you about the mantis shrimp eye, I want to tell you what a typical crustacean eye looks like so that you can understand the complexity that we find in stomatopod visual systems. So start off with this Hemigrapsis crab. They have compound eyes. We have camera-like eyes with a lens and a retina at the back of our visual system. In crustaceans, they typically have compound eyes. Compound eyes, think of a fly eye. It is made up of individual units, and each unit is called an ommatidium. If we take a look at one of those ommatidia, pull that out and blow it up, each ommatidium is made up of several components. There's a cornea and a crystalline cone. So these are optical structures that help focus the light, and they focus the light onto a set of receptors underneath. Crustaceans typically have two types of receptors. They have a UV receptor, which is a single cell that sits on top of a blue-green receptor, which you'll also hear me refer to as the main receptor. But they have two receptor types. That main receptor is composed of a ring of cells that project microvilli into the middle. They basically create a tube that they put a light-sensitive protein in, and that light-sensitive protein or visual pigment is what's responsible for capturing wavelengths of light. So one thing to take note of is that in most animal species, there is one visual pigment that's responsible for the blue-green sensitivity of the main receptor, and there's another visual pigment that's responsible for the ultraviolet sensitivity of that UV receptor. So for every spectrally distinct receptor, there's a different visual pigment responsible for that. So that's how a typical crustacean eye is put together. What about stomatopods? So stomatopods are, because they're predators, you can imagine they're highly visual animals. This video shows you that they've got a pair of compound eyes. It's up on stalks. They can move them around independently to look at different things. If they're particularly interested in something, they will take both eyes and look at that. But otherwise, if they're just looking around the world, the eyes are going to be doing different things. If we take a closer look at just one eye, there are a couple things that stand out. First, if you look at these black regions, these are called pseudopupils. So these are the parts of the eye that are focused at the observer. So by the fact that there are three different black regions in the eye, it means three different parts of the eye are focused on one point in space. So we have binocular vision because we have two eyes. They have trinocular vision with one eye. And that has to do with the fact that they have a set of these ommatidia, 6 rows of them that run around the equator of the eye. And packed into those 6 rows of ommatidia are a set of receptors that are specialized for different visual tasks. If we look at a schematic, so this is similar to that crab set of ommatidia that I showed you. So the crystalline cone and cornea here, light is coming in from the side. Each one of those colored structures is a different receptor type that you find in the stomatopod eye. And we have numbered here the 6 midband rows. So you can see by the different colors and the different shapes and structures of those receptors, they have this set of highly specialized receptors in those midband rows. I can further characterize what's going on there. If we run through each receptor, we can characterize each by the sensitivity, so what wavelengths of light is it sensitive to, and then also by the visual task that stomatopods use that for. In the hemispheres of the eye, and I have up here an eye diagram that will be colored to tell you what part of the eye I'm talking about. So if we start in the hemispheres of the eye, those main receptors, so here's a sensitivity curve for those. They're most sensitive to about 500 nanometers of light, so that's blue to blue-green. And these receptors are responsible for basic spatial and motion vision. If we move into the midband rows and look at the first 4 rows, those main receptors have now been subdivided into 2. So there's a set of 6 receptors now. Each one is sensitive to a different wavelength of light. Those 6 are spread out across the visible spectrum, so you have fairly evenly. So you have red all the way down to violet, represented by those 6 receptors. And these are thought to be the color vision system for the stomatopod. In rows 5 and 6, that main receptor has a single sensitivity, again, to blue-green wavelengths of light. But this set of receptors is specialized for the detection of polarized light. So they can detect the plane that wavelengths of light are vibrating in. And then finally, those single cells of ultraviolet sensitive receptors that sit on top, there are 6 different types of those. Altogether, there are 16 spectrally different types of photoreceptor in the stomatopod eye. So to give you some perspective on how unusual this is for an animal visual system, pod set of receptors compared to our color vision system. So we have a trichromatic color vision system. We have a blue, a green, and a red receptor. And with those 3 sets of receptors, the diversity of colors that we do, that's a very large diversity. Stomatopods see 16 different distinct colors. So in the field of understanding stomatopod visual systems is what in the world are stomatopods? I mean, if you calculate out what we see with 3 receptors, what stomatopods might see with 16, it's something you can't comprehend. There's another piece to this puzzle as well. As I mentioned with the crabs, they have 1 visual pigment per distinct receptor. And that's true if you look at most animals. So we have 1 visual pigment for blue, 1 for green, and 1 for red. Some other animals, so some birds have as many as 5 receptors, and they have different visual pigments for each of those. So stomatopods have at least 3 times more than any other animal that we know of. The diversity of colors that we do with our visual system, we have 3 receptors and we have 3 opsin proteins. So those visual pigments, there's a distinct one for each one. My main area of research has been looking at the molecular evolution of these visual systems. So how many different visual pigments are expressed in stomatopod eyes that go along with these 16 spectrally distinct types of photoreceptors. We hypothesized at the beginning there would be 16. There are 16 receptors. It made sense there'd be 1 visual pigment for each of those. What we found is that there are at least twice as many visual pigments expressed in the stomatopod eye as they need. And we're finding really unusual expression patterns. So some receptors will express as many as 8 or 9 different visual pigments. And visual pigments that we think have different spectral sensitivities. So stomatopods are doing something not only unusual with their photoreceptors, they're doing something really unusual at the molecular level as well. And that's a big question in my research program now, is what are they doing with all these visual pigments? And how does that translate to a functional eye for the animal? One question is how they are processing all of this information. So if we look at the way that we see all of the colors that we can perceive with our 3 sets of color receptors, we're able to see many more colors than we can detect because of our big brains. So we're able to process that information and compare the input from multiple receptors. So if we look at our eye, this is our retinal layer. We have photoreceptors here, our blue, red, and green. We have an additional receptor that we use in dim light, we call rod receptors. There are multiple layers of ganglion cells below that integrate the information from those photoreceptors. So 2 photoreceptors here get compared in these bipolar cells. The input of multiple bipolar cells get compared and summarized in these ganglion cells. And all of that happens before any information is sent back to the brain. So we're able to see yellow, for example. Even though we don't have a yellow receptor, we can compare the input of our green and red receptors, and our brain will perceive that as yellow, even though we're not specifically detecting it. So what's going on in stomatopods? Like I said, for a long time, there was this idea that if they were integrating and processing information the same way that we were, they should see exponentially more colors than we can detect. The first person to really test this was a friend of mine, Hannah Thoen, at the University of Queensland in Australia. And she did a series of behavioral tests. Stomatopods you can train to associate a particular color with food. And so she did choice tests. She would train a stomatopod to one color, and then she would give it a choice of two different colors. If the stomatopod can tell the colors apart, it's going to go to the right color that it was trained for. If it can't tell the colors apart, then its choice will be 50-50. So it's going to make a lot of mistakes. So she did this to ask what the spectral discrimination of these animals are. And by spectral discrimination, I mean, how far apart do two colors have to be before the animal can perceive them as different colors? So if you look at this graph, let me walk you through it. So we have here different wavelengths from short, so ultraviolet down here, red on the 700 nanometer range. On the y-axis, we have difference in lambda max. So what is the difference in wavelengths? How many nanometers apart do two colors have to be before an animal can discriminate them? So it's a little bit washed out, but there's curves here for a honeybee, a butterfly, and the human. And the lower that curve, so for the honeybee, the closer two colors are, the better it is at telling that color apart in that part of the spectrum. So here the honeybee is very good in sort of the blue-green range and discriminating colors in that range, but it's not so good down here in the violet. And so you can look at these curves and you can tell where animals have really good discrimination. They can tell two colors apart that are very close to each other, or very bad color discrimination. If you model the stomatopod visual system based on how our visual system works, it should have great color discrimination. So it should be down here below humans and better, and certainly in longer and shorter wavelength part of the spectrum than the other animals that have been measured. Through these choice tests that my friend Hannah did, she found that they were actually really bad at discriminating colors. So this is counterintuitive. Nobody predicted this. So what you find is that black line on the top is the color discrimination ability of Haptoscola trispinosa, the species she was working with. And it is worse at discriminating colors than any other animal tested. So you have to get to 10 or 15 nanometers apart before the stomatopod can tell that there are two different colors. So why is that? And I think that the best explanation, and this is the biggest question in stomatopod studies of stomatopod visual systems now, is what are they doing with the visual information? So what this suggests is they are not comparing the inputs of photoreceptors like we do with our visual system and most other animals that have been looked at do. So they're processing the information from that set of photoreceptors very differently. And what Hannah and her collaborators, Justin Marshall, proposed is that rather than comparing inputs to be able to discriminate colors, their visual system is more of a frequency detector. So if you give the stomatopod visual system, here is a blue signal. So if they're looking at something blue, this set of receptors gets stimulated, and the brain says there's some threshold that says a bunch of blue receptors are stimulated, that color must be blue. Similarly, if the stomatopod is looking at something red, represented by this line, this set of receptors gets stimulated, and a signal in that part of the brain says, I'm looking at something red. So stomatopods can see blue from red, but they're pretty bad at telling red from red-orange because of the way the information is processed. They have tiny brains. So a bit of it may have to do with constraints of the processing power that they have. We can do all that processing with our brains. They have very small brains, not as much processing power. Another feature that may contribute to this, again, human versus shrimp and visual information processing. So if we go back to the schematic of the human retina, there are photoreceptors here. Multiple photoreceptors get compared at the bipolar level and integrated as a signal. Multiple bipolar cells take information and that gets integrated into ganglion cells. And then that information gets sent back to the visual cortex in our brain. And then that information gets parsed out and sent to centers that tell us things like, what is it are we looking at? Where is it located? So it gets highly processed before it gets sent back to the brain. In shrimp, we're looking at this part of the mantis shrimp. The eyes and the brain is located right here. So if we look at a diagram of the wiring and the neurons that project from the eyes, here is that midband with all those photoreceptor diversity that we just talked about. What Hannah and Justin Marshall and their colleagues are proposing is that rather than all this processing that you see happens in our visual system, receptors are kept separate. So it's a parallel stream of information. It's parallel processing. Each receptor gets its own channel back to the brain. So each one of those gets sent back to the brain. And it's in the brain that then is detecting what sets of receptors are stimulated. Advantages to this system is that it's very fast. It's a direct channel back to the brain. All that processing takes time. So one idea is that perhaps it has to do with the processing power of the shrimp relative to the speed of its predatory strikes and capturing prey. But these are all questions that people are actively researching now. And this is, like I said, is the next big question. We know that based on Hannah's work, the information is not being processed similar to color visual systems in other animals. So the next question is, how is it being processed? And they're really trying to track that down now. So what you have again is this frequency detector. And other sensory systems that work more like frequency detectors are our ears. So what we have in the stomatopod visual system, the best current hypothesis anyway, is that they're looking for sets of receptors that are going to be stimulated above some threshold to be perceived as a particular color. And because of that, you can't have very fine discrimination. So with that, I hope that you've learned a little bit about vision in the ocean, and particularly about how bizarre and wonderful stomatopods are and how unique their visual systems are. There are still a lot of questions to be answered, and there are many mysteries left to be solved. I wanted to just quickly thank the group of people that I work with. It's a colorful group. They study vision, so they like color a lot. I particularly wanted to point out Roy Caldwell and Mike Bach. All of the beautiful pictures and videos, or most of them anyway, in my talk are not because I have any skill, but because they're really fantastic photographers. And then, of course, all of the sources that have given me money to do my research, including the University of Hawaii. And with that, I'd be happy to take any questions. Thanks. Yeah.
You're looking at one particular stomatopod. Do all the stomatopods have the same visual structure, or does that depend on where they live, what they do for a living?
So that's a really good question. The question is whether or not, so I presented to you one set of receptors for stomatopods, and the question is whether there's variation in visual systems among species of stomatopods depending on where they live. And in fact, what I've shown you is the most complex visual system that we know about in stomatopod species, but there are many species that live, for example, in the deep sea, or at least in darker habitats, deeper in in the ocean that have lost some of that complexity or don't have any of those mid-band rows at all. So even within the group of stomatopods, you have this really fantastic variation of complexity in the sets of photoreceptors that they have.
Two questions. What are you using to determine all this? Is it all microscope work? And the second one is, did this evolve because they're down where the light source is very poor down at the bottom of the water?
So the first question was about the techniques that are used to determine some of the features about the visual systems. And I wanted to reiterate, a lot of what I presented to you is sort of 20 years worth of work, doing sectioning, so looking at structure and ultrastructure, so microscopy work, doing physiology, so doing electrical recordings of responses of different receptors. So you can flash light, different colored lights at receptors and see if they respond. You can measure directly the absorbance of the photoreceptors. A lot of my work is molecular evolution, so I look at the evolution of the DNA, the gene sequences and the proteins and how those are functioning, where they're expressed in the eye. The second question was whether or not this evolved deep in the ocean. And for stomatopods, I would say roughly 2/3 of the species live in shallow water habitats associated with coral reefs. So they have this diversity because they live in colorful environments. Well, right, so they're living someplace where there's a lot of color and a lot of features available to look at. So a lot of this diversity evolved in shallow water habitats. Where you see less complexity, it's because species have probably moved into deeper ocean waters. Yeah.
Question is, what do they use the color for? You have offense, finding food,
Mm-hmm.
defense, protection, and sex, finding a mate. Now, for offense, the ones I studied years ago when I was a lot smarter, color isn't important. Size, shape, and speed, especially the punching ones. Giving the opportunity of several different foods that are really slow moving. They seem to pick the one where they can get the most food for the least amount of punching.
So the question is, why do they need to see all this color? And the three basic reasons that animals need sensory systems, and vision in particular, is for mating, for finding food, and for avoiding being eaten by something else. And so the answer really is they use their visual system for all three of those things. By having trinocular vision in one eye, so they can range find, and they need to be able, because of this very fast strike, they need to be able to target something precisely. So some of it may be related to their predatory lifestyle and that really fast strike that they have. They're also, as you saw, many of them are very colorful species, and they have a lot of communication between individuals for mating, for example. For avoiding being eaten, there are things that eat them, so I imagine they're on the lookout for that. Octopus will eat them, some fish will eat them. So they're using different parts of their visual system for all of those tasks. There was a question over here.
Because of how their optical systems wired,
Mm-hmm.
wired directly to the brain,
We think. They're still pinning that down.
is their optic nerve more like fireballs versus the human body? I mean, how big is the...
That's a good question. So there is still some processing. So I sort of glazed over that, but they're in the eye stalk. There's 3 centers of processing that any information from the visual system goes through. But each one of those receptors is, the signal from those receptors is kept separate, at least through the first 2 sets of neuropiles, the first 2 processing centers. So a lot of the work now is concentrating on trying to track that receptor neural connection all the way back, and to see how far back it's kept parallel, or is it integrated somewhere farther down the line? So those are still open questions. We don't know if it's a fire hose. It's not, I don't think, a particularly big neurons we're talking about. They're not particularly big animals. I have no idea if it's unusual for a crustacean or not, but that's a really good question. We don't know. Yeah.
So they have visual. What else senses do you guys know about that they have?
What other kinds of senses besides vision that mantis shrimp have? So they have very good chemical sensation, chemosensory. So if you've ever seen a live stomatopod, they do a lot of antennal flicking. So they have chemosensors on their antenna, as many crustaceans do, and so they will taste the water. And there have been behavioral studies that show that they can identify individuals that they've fought before. They can identify by smell alone. So they do pay attention to chemical cues, and I'm sure it's involved in mating, although I don't know of any specific studies that have tested that. So they have chemical sensing. Some of the species, the big Lysiosquilina, as well as a big species on the coast of California called Hemisquilla, they make a low frequency rumble, so they can communicate by sound. We're not exactly sure how they're detecting that, or at least I'm not. The person who's studying it may have a better idea of that. So they can use, some species are big enough to produce a low frequency sound that will travel through the water to communicate with other individuals in the area. So they use a number of other senses as well. Yeah, in the back.
These invertebrates, some of them can regenerate appendages through trauma or predators. Can the eyes be regenerated?
That is a great question. So the question is whether or not eyes and stomatopods can regenerate. Since they're such an important sensory mode, they're obviously very visual animals. So if they lose an eye, one good thing about the way their visual system is set up is with trinocular vision in one eye, they still have depth perception if they do lose one eye. It turns out if they lose most of an eye, rather than growing back an eye, they grow back an antenna. So they don't seem to be able to regenerate an eye. Why they make an antenna instead, I'm not entirely clear. It's an interesting question that I would love to have someone play with in my lab, but it seems a little cruel. So yeah, they don't seem to be able to do it. Yeah.
Have you ever been attacked by one yourself?
Only when I am handling, when I'm catching them and they're obviously disturbed, then yes, I've been hit by one. Both. But never by. I'm very careful with the big ones because the big ones can do serious damage. So we take extra precautions with those. But they're not generally going to come after you unless you mess with them first. Yeah.
I seem to have missed the thing about the hearing. You said they hear with their eyes. That was the title.
So the analogy is that because they're processing the visual information very differently. They're not processing the visual information like other animals. So they're, rather than detecting sort of specific wavelengths, they're looking at the frequency of wavelengths that are being detected, which is more like an ear works than an eye. Well, what we know about other visual systems anyway. Yeah.
You keep them in plexiglass tanks or glass tanks?
So only the very, very biggest ones can break tanks, and it's extremely rare. We don't have any here that are big enough to do that. So I don't worry about it too much.
I had one cracker tank.
Really? I would like to know what kind that is.